Dzień dobry Państwu.
Hello, ladies and gentlemen. My name is Katarzyna Mucha, and on behalf of the Management Board of Molecure, I would like to welcome you at the quarterly meeting after the publishing of the financial report for this quarter of the year. The Management Board. There are some technical issues. I'm sorry for that. The Management Board will present the update on the progress on the clinical and preclinical projects and the discovery stage projects, and will sum up the business development activities and the operational and financial standing of the company. The presentation will be given by Marcin Szumowski, the CEO and the Chairman of the Board, Piotr Iwanowski, Chief Medical Officer and member of the Management Board, who took the office on the 30th of September this year, Zbigniew Zasłona, Chief Scientific Officer, and Sławomir Broniarek, who is the Chief Financial Officer. The meeting is planned for 90 minutes. We have planned for you two language versions, the original Polish track and an interpretation into English. First, we will hold a presentation and then a Q&A session. Please send your questions via the chat. Thank you, Kasia. It is our pleasure to be able to meet you again after the holiday season. Today, we will be talking about the results for the second quarter of this year. The company's objectives, strategic objectives, remain the same. However, some new things have happened in the last half year, in the last few months. Mostly, I would like to say that the chief study which we are currently conducting is the ATD-01 study on patients with lung sarcoidosis. We are currently recruiting patients for that study, and new publications, new information which appeared over the last half year are data regarding liver diseases, so MASH. And here we will be able to show new things in this area, and we will tell you why this market, from the scientific perspective, genetic perspective, and market perspective, is an incredibly attractive one, and what ATD-01 can do in treating this disease. The second clinical project is ATD-02 in immuno-oncology. Here we are in the course of administering the fourth dose to patients, and more about this will be discussed by Piotr further on in our presentation. The two other areas which we will be elaborating on is the platform for discovering new targeted drugs for mRNA. We have made progress in this area as well, and we are starting two individual drug discovery programs among the targets which we analyzed in our platform. And USP7, we are currently waiting on results of our collaboration with an American company, Avicenna Biosciences, which is to utilize its AI tools to improve the parameters of our most advanced candidates. And as Kasia already said, we would like to welcome among us Piotr Iwanowski, who will take over as Chief Medical Officer, but also member of the Management Board. He is not a new person for our company. We have been cooperating for 5 years already. However, now we are intensifying the forms of our collaboration. When it comes to our pipeline, there is not much new. We'll be discussing more about where we are with each study and which activities we are carrying out. However, what we'd like to say is that we are focusing our resources, our finances on the clinical trials, and the progress in early studies is also being made. However, currently Unfortunately, we are less actively involved in this area when it comes to the company's finances. The clinical trials are the main driver for the costs, but it's good that the costs are there because it means that we are progressing forward. Now I'd like to pass the floor to Piotr because he could tell us more about the KITE study.
Good morning, ladies and gentlemen. The KITE study is not something new to our listeners, I hope. It is a phase 2 study, so it is a study in patients already who are ill. It is against placebo. It is not a very long study, so it is bioethically and medically feasible, and with the indicators that we've adopted, we want to prove the efficacy of the compound. We are now still at the setup stage, so not all sites have been activated. As you know, the US was the first to be activated, the US sites. Now we are initiating intensively and activating the last site to be able to enroll patients in the site as well. So looking at the countries, we are already operating in the US, UK, EU and Norway. We have 13 sites initiated at the moment, and we want to activate 4 more sites within the coming weeks. This study is, we are also applying for a new grant to the National Institute of Health in the US for $2.2 million US dollars. And in terms of the conduct of the study, as you probably know, it is conducted by the CRO, by a commissioned clinical operator, but we as a sponsor try to actively make the study visible to facilitate the actual enrollment of patients, so it is optimal. Currently, the pace of enrollment is not satisfactory for us, and we hope it will change for several reasons. First reason is that we will start the study in Europe, which, from what we know, is a continent where the patient enrollment is easier. Also, we have seen that there is more room in the clinical trial landscape at the patient level, because there has been a study that has finished recently, a Dutch academic PREMAT study, and also we have seen a comparable study, a competitive study, in terms of resources, patient resources at the sites, the so-called Resolve Lung study, which was conducted by Kinevant Biotech company at a number of European sites. So these two facts, with the new sites opening, we make us optimistic as to speeding up the enrollment of patients. However, even when cooperating with the best CRO company, there are a lot of activities still remains with the sponsor, and this is my priority as the new person on the board, my priority to act as a sponsor, which is to improve the visibility of the study among doctors among experts, to improve also among other doctors that are not our researchers, but the local physicians, the referral rate, and also the visibility at patient advocacy organizations and sarcoidosis-related patient organizations, and also to improve visibility for the patients themselves. On this slide, we have summed up the scope, the outline of our strategy of improving the enrollment of patients in the KITE study. So some of these aspects I've already mentioned, but what I would like to start with is that pulmonary sarcoidosis is by definition a rare disease. It's sort of at the verge of the definition of a rare disease, and in terms of epidemiology, but our study is not addressed at all the patients, but only those that are treatment naive, that have not yet been treated for sarcoidosis, but they are eligible, they qualify for treatment, or those patients that have not been treated for the last months. So there's such a subset of patients is a result of the need to have this placebo-controlled group, which is a necessity to prove the efficacy of the drug at endpoints. So in terms of competition, I've already mentioned the two competitive studies that have been completed. Also, we have our strategy ready for improving enrollment. This strategy consists of a sort of deeper screening of. We have reformulated the strategy in September. The European Sarcoidosis Congress was an event in which we participated, and we had a number of meetings with researchers, with potential researchers from other potential participant countries that we are considering as our Plan B or Plan C, also with patient advocacy groups such as the Sarcoidosis Foundation from the US. And we have identified the following fact, that in the US, the approach to the need of the enrollment needs is different than in Europe. So in the US, there is a need for more visibility for the patient themselves, whereas that is able to be in direct contact with a CRO, whereas in the EU it is more important for the study to be visible to doctors that can issue referrals for specific patients, and also to patient advocacy groups. So our strategy is sort of different in each of those two areas. So all local conferences, events where doctors are attended by doctors that will be also attended by us with our project. And we also intensify activities to identify an optimum site, optimum country for backup countries to expand our study, but we do not yet see it as a necessary measure.
And now I am giving the floor back to Marcin. Despite of the fact that we are currently carrying out our trials in sarcoidosis, another indication which is much greater in terms of the potential patient population is MASH. And we are talking not just about the number of patients, but also the growth dynamics of the potential market, which results in the fact that it is the largest market for the potential use of OATD01 right after gut diseases, where we also are receiving a positive in vivo study results, which will this will account for half of it in 2032, and right after lung diseases, which are a considerably smaller market. But the market size is not the only reason why we decided to focus over the previous year to analyze the pathogenesis of the disease and our therapeutic objective. So, type 1, which intends to stop or reverse the progression of MASH. Here we are presenting the growth dynamics of the market, over 40% of growth per annum, which will lead in up to over $25 billion in 2032. That's the time when our drug might have already hit the markets. And this is one of the main elements of our strategy. As you probably know, we have one drug approved. This drug has been approved only for early stages of MASH, so stage 1 and stage 2 of MASH. We will present in a moment results which suggest that our molecule can also be effective for far more advanced stages of the disease, when we are experiencing fibrosis and the change of the tissue in the liver. And here I would like to pass the floor again to Zbyszek, who will tell us about the preclinical projects. MASH is a disease of the liver, is a hepatic disease. Traditionally, in our company, we focused on the lungs. However, the results which suggest that inhibiting the activity of chitinases is desired in inflammatory diseases affecting other organs, not just the lungs. This is something we have known for a while. But over the last year, we have been working on a MASH model, mostly because we wanted to learn the mechanism of action of our compound on the molecular and cellular level. And at the same time, we received more and more data from patient biopsies, and also the omics study, which without any hypothesis tried to specify what are the main factors causing the disease. And here there were a few things that happened at the same time. Marcin presented the statement of reasons for our decisions, and now I would like to present the scientific basis for our actions. In the top of the slide, you can see a model of the MASH disease, and we compare with the ALK5I compound. It is a classic inhibitor, a classic antifibrotic drug. As you can see, Cyrus red staining, which presents the level of lung fibrosis, shows us that a traditional antifibrotic drug has a similar action to ours. However, when we look at the panels below and we see the staining on CD68, the marker of activated macrophagy, we can see that our compound reduces the level of macrophagy activation and its number, while the reference compound does not achieve that. This shows us that this is a new way of solving the problem of fibrosis, different to the traditional one, which was not particularly successful. When we had a closer look at what happens after we administer our drug, the main conclusion we came to was noticing the change of metabolic processes on the cellular level. To the left, you can see the list of biological processes after the IT specialists have processed the data typical for MASH. And on the right-hand side, you can see what happens to MASH after OATD01 has been administered. And from top to bottom, you can see the most activated processes into those which are inhibited. The processes in bold are the ones which have been inhibited in the course of the disease, and this is reversing of the progression of the disease in this regard after the administration of our compound, and that is what we are focusing on. One of the processes is the process of glycolysis, which has direct use in the PET scan reading, because it shows how preclinical data can help in reading preclinical trials. So PET-CT readings is about the absorption of glucose and determining the inflammation. We hope that OATD01 will reduce this. So what we have learned with this study is that OATD01 can change metabolic processes on a cellular level and therefore have a therapeutic impact on the course of MASH. A week ago, there was a publication in a good journal which characterizes HIT-NS1 as a therapeutic target. The authors of this publication have carried out a very elegant set of preclinical experiments where they presented that by using our OATD01 inhibitor, they are able to inhibit inflammation, hepatic inflammation, but also fibrosis in the MASH model. And at the same time, they collected samples from patients with MASH and fibrosis, and they have demonstrated a perfect correlation of inflammation, which ends in fibrosis, with HIT1. So, as the article shows, HIT1 is not anonymous anymore when we are talking about MASH. These are data which we took from a website devoted to patients with MASH, where various clinical trials have been collected, trials which use anaerasic methods for expression of biomarkers. Marcin already mentioned that MASH can be divided into 4 stages from F1 to F4, where F1 means minimal scarring caused by inflammation, F2 means scarring with signs of fibrosis, stage 3 means that fibrosis can cause significant macroscopic changes, and F4 is the final stage of the disease. You can see probably how great the HIT1 expression correlates with the disease progression, and this data comes from a large sample of patients. That was 81 healthy patients who were F0 and over 500 patients diagnosed with MASH. MASH, there is more literature suggesting that MASH has clinical significance. This is an example of a different publication which discusses the HIT1 and MASH, so HIT1 expression. MASH is frequently correlated with type 2 diabetes, and HIT1 can also serve as a marker which is correlated with insulin resistance. The next slide will be taken over by Piotr, who, I don't know if you are aware, is also a geneticist by education, and he will be able to tell us more about this project. So he is a doctor, a medical doctor, but he's also I'm also a genetician. Apart from carrying out clinical development studies of drugs, I do other things as well.
So these studies are quite similar to the ones mentioned by Zbigniew. From a decade ago, an academic study in which a status of activity of chitinase was a phenotype that depends on the genotype is related to the risk of steatosis and its gravity. So, as some of you know, probably the KIT1 gene that is encoding the protein that is the molecular target for our drug has a fairly popular sort of common mutation, which if it appears on both alleles, then the KIT1 activity is practically limited to minimum. This part of population corresponds to a patient that takes our drug. So a very simple method, but a very spectacular study is a clinical case-controlled study in which they compared and they looked at the correlation between in 200 patients, 110 patients were patients with NASH, 90 patients had simple liver steatosis, and 100 patients were a control group. So this, of course, is 300, not 200 patients, and the yellow elements is the genotype, where patients that have the mutation of both alleles, and so the person can be either healthy or this person can be in the group with liver steatosis, and you can see that very clearly that this mutation has a protective character on the chemical phenotype of the patient. So patients with this mutation out of the group of sick patients do not show the symptoms of the disease. So the patients without the KIT1 activity do not have the disease, and you can also see how this number of 110 patients distributes over the gravity of the disease over the scale of fibrosis of liver. And here you can see that the genotype where the KIT1 activity is 0 has a protective sort of nature. So it correlates with a smaller risk of fibrosis. Almost no patient in this study, no such patient developed heavy fibrosis. So to sum up, if we were to look at another indication for our molecule, which is MASH, this is our own data from preclinical studies and from literature from this clinical case study, we have serious reasons to consider MASH for further development, taking into account also both the epidemiology of the disease and the potential and the market potential, because in this disease, the first drug has appeared only recently. So this was the OOTD-01 development. Now I would like to move to OOTD-02 clinical study. You probably know very well that we already are in phase 1 clinical study. However, it is already in oncology patients, so it's sort of between phase 1 and phase 2. So this is an open label rollover study. So patients who complete 6 months of treatment in the study and they can and want to continue have a possibility to participate in a separate rollover study. We operate in 3 sites in solid tumors of colorectal cancer, the ovarian cancer, pancreatic cancer, and also renal cell cancer. And our drug is given as monotherapy for patients who do not fare very well and they do not have an opportunity for any standard treatment. So each patient receives the drug for 6 months. They receive they receive the drug until any signs of dose-limiting toxicity appear. So which would show that perhaps there is a need to limit the dose to receive the so-called maximum tolerated dose in our study, but it's still ahead of us. We are now at the 4th level out of 6 escalation levels. So we are at the level of 20 milligrams per day, and 3 patients have already completed the observation period. So the first month in the study. And at this level of dose, we have not observed any DLT, so no adverse events that would, according to the protocol, would need to be considered directly as adverse events leading to modifying the dose escalation. So we can. So therefore, we have this flexible model where it would actually signify de-escalation to smaller doses. So after the 3 patients have completed the DLT observation period with the 20 milligrams per day, we have had a routine meeting of the safety review committee, an external independent committee that looked through the safety data and also pharmacokinetic and pharmacodynamic data on the drug saturation in the body of a patient and on chemical effects in patients, and they have recommended for the treatment to be stopped at the 20 milligram per day and to collect the data from the next 3 patients. It has not been caused by any DLT events, but it has been dictated by the variability of the pharmacokinetic and pharmacodynamic results in different patients. So the group of 3 patients, of course, in biomedical studies is a very small group. So it is not a group on which you could draw binding conclusions. If you see different sort of results, there is a possibility of having outliers. So perhaps we have patients that for some reason have shown higher or lower results than standard or typical patients in their biochemical effect. So in order to see whether our patients are lower or higher on the scale, we have the committee has issued such a recommendation. Of course, we comply with the recommendation of the committee. So now the recruitment of 3 more patients is underway, and the information that we've received that today we have another patient. Only today we have another patient starting its treatment in one of the sites.
That's it when it comes to the clinical part. Thank you very much. And when it comes to the preclinical projects, Marcin already mentioned our cooperation as part of USP7, our cooperation with the company Avicenna. On the slide, you can see what this cooperation is about. For a few years now, on working on our USP7 project, we have managed to generate quite a lot of data on the molecules which have been synthesized and studied by us. And Avicenna specializes in a situation like this, where we already have a data set on which you can work. We have some crystals, we have some molecules which have been already described, not just with, in terms of affinity, but also their drug-related functionalities are well described. And Avicenna, using their algorithm, will be able to handpick the right molecule which is most promising. This is the objective of our cooperation. As you can see in the slide, this is cooperation which consists on a shared risk and a joint venture, which is supposed to bring us a good candidate rather than a fee-for-service arrangement. When it comes to the administration route, we are looking for an intravenous solution. Here we have candidates. We have a lot of faith in the USP7 target, and therefore we have increased our activity on different fields. So intravenous administration and oral administration. When it comes to IV, we are considering strengthening the pharmacokinetic properties of our product by packing it into liposomes, which would help optimize the properties, the drug properties of the compounds. This is a situation where in a cancer indication, this is a natural form of administering drugs in this cancer indication, and that is also justified from this perspective. When it comes to the mRNA platform, as Marcin already mentioned, after the 2 successes where we have managed to prove proof of concept on a cellular level, where we managed to prove that the molecules which we have developed have inhibited the proteins, we moved on to adding context to those molecules. So here I would like to tell you in which context they are being developed as we speak. To the left-hand side, you can see one of the most promising compounds, which inhibits the translation of protein, which is dose-dependent. And to the right-hand side, our compound, when administered in concert with a known apoptosis activator, has a synergistic effect, a very strong one. It is a situation where we are able, by using those two compounds, we are able to simply kill cancer cells and inhibit tolerance against drugs which otherwise wouldn't work. Currently, we are at the level of synthesizing compounds which would be more selective and which would work, which would be effective in smaller concentrations. Another program is a program which we are developing in the context of inflammatory diseases. The first hit in this program was derived from our cooperation with a CRO company, which specializes in identifying molecules in mRNA by way of a DEL screening campaign. This first hit, which we've achieved, was characterized on immunological cells. And as you can see on the middle graph, this is dose-dependent compounds inhibit the secretion of pro-inflammatory molecules without affecting mortality. This is the desired effect. This is what we wanted to achieve, and currently we are thinking about autoimmune inflammatory diseases, where the greatest benefit would be experienced by patients who currently there are antibodies, but a small molecule would help them receive treatment without an inpatient setting. They would not need to be hospitalized. They could receive the treatment in their in-home setting. So this is another program that we are leading. It is at quite an early stage. As you can see, it's quite innovative. Two more things regarding the mRNA platform. First of all, we are developing a molecular test, a molecular assay, which we would use for screening of the first molecules, and that test we intend to patent. This is a situation which would provide our mRNA platform to be unique in terms of this approach. The next thing is that we are developing our AI approach with regards to foreseeing the areas which can have great affinity with the compounds. So simply identifying druggable regions. These are the works where the mRNA platform is being moved forward into development. And now I will give the floor to Sławek.
Thank you, Zbyszek. After this scientific data, we would like to show you how much it has cost us. On the left side, you have the profit and loss account. So the loss compared to the last year has been higher by 3 million, which is a question. So what's with the revenue? Because we miss some of the revenue from grants that we had last year due to the fact that because we have also lower level of funds on our accounts, we have less interest on that. We have a grant in RNA so the use of grant in RNA is zero. Why? Because the Polish Agency for Enterprise Development has failed to settle the payment claims that we have submitted to this institute institution, to this agency, we have 2 million zloty of due payments. But other than that, the company has decreased costs by 20%, making some savings in external companies in related to IR and consulting services. So at the cost level, we try to keep the EBITDA at the similar level. How does it look like on the balance sheet side? So we see a decrease of 26 million zloty in the balance sheet, which results from the change in value of our clinical projects, changing the accounting policy. We only capitalize the clinical projects, and the funding. So you can see here the 14 million are the costs from projects other than the clinical stage projects. So in terms of cash flow details, we can see that there is the same dynamics year on year in terms of spending on clinical and early stage projects. The data is very similar year on year. We have spent around 10 million zloty on clinical stage in the first half of the year, just like last year, and we try to limit administrative costs to minimum, make them fit them to the company's situation. So both last year and this year we have spent approximately 26 million. The cash position is approximately 27 million zloty, and we have had around 113 employees. Over 50% are PhD level employees. So now I would like to move on to some of the sources of our funding. We would like to have more dynamic funding, but as you know, the recent decisions of the PARP, the Polish Agency for Enterprise Development, have not brought us closer to this result. From the projects for which we've obtained funding, there is the mRNA platform. We had a positive decision. There's been some success in this platform, but the OATD-01, unfortunately, the application were rejected. Waiting for evaluation, we want to submit a protest and appeal to the OATD-01 against the OATD-01 agency's decision. We expect the NIH, the result of the NIH decision. Here we are quite optimistic, and also we have just completed a panel at the National Center for R&D related to a grant for the implementation of artificial intelligence tool in the discovery phase for our protein and RNA target drugs. And we have a tiny project worth some 5 million zloty related to the validation. Another validation, and Zbyszek will tell us more about it, validation of our potential of OATD-01 in MASH disease, which is performed in consortium with the Swiss Technical University. So this is everything as far as grants, subsidies are concerned, and this is a summary of our plans and sources of funding and our expenses. So by the end of last year, we plan to spend some 60 million zloty on clinical phase programs. So how much do we have left to spend? Some 2/3 of this amount, 35 million for preclinical RNA, 25 million, and 28 million for administrative and other general expenses. We have a cash position of some 27 million. We look for non-dilutive grants. We have we have some funding already granted under the mRNA platform, and we look for AI grants and for other types of grants. The main sources of funding should be partnering transactions and also funding from the capital market, where this should make up for the difference, which is approximately 100 million zloty. Thank you.
Thank you. So, to sum up also a bit about business development, because this is what we spend a lot of time on, and this is where we have a lot of interactions going on. Those numbers, these figures that you can see on the slide indicate where our main focus is and where the main interest is, where the discussions are most advanced, and also where we would like to firstly monetize on the results of our research works. All those parameters focus quite clearly on OATD01. This is the number of meetings from the last Bio International, after which we had a lot of follow-up activity going through subsequent stages. But here our priority, which results from various of the genetic scientific market results and the growth dynamics, and the clear picture of the data, as told us by Piotr, in the preclinical aspects, which come from both our laboratories, in-house laboratories, and also from Galapagos, as indicated by Zbyszek. All this points to the fact that the key target is MASH. So the macrophagy, and by way of modulating the functions of those macrophagy, we affect the course of the disease. And at this point, I would like to underline that this is something which is the target, which a Polish company, and the mechanism of action, all that is novel, all that is new. A lot of companies are quite cautious about these potentially breakthrough therapies because they have not been validated, because this is not something that everyone knows, as in the case of inhibitors, control point inhibitors in cancer care, which is also a breakthrough in the existing cancer care therapies. They came in more than 10 years ago. They helped cure some of the cancers. And here we are dealing with a potential breakthrough in the treatment of autoimmune diseases, inflammatory diseases, which change the structure of tissues and which work through our target, that is modifying the macrophagy phenotype. This is where we want to focus our attention when it comes to business development, because this is a potentially huge market. And potentially the breakthrough would be great, would be huge. Of course, the talks that we are carrying out involve also the other clinical program, so OATD02. We had a lot of mRNA meetings, mRNA-related meetings. And when it comes to value driving and the potential cash that might inflow to the company, definitely OATD01 has the largest potential in this regard in terms of potential revenues. Generally, our pipeline is perceived very positively, is perceived as something innovative, as something first-in-class potential, on the other hand, and this is quite typical for this sector, and even more so in the post-COVID area. A lot of large companies such as Pfizer, Merck, Lilly, Novo Nordisk, those huge whales of companies which have a strong market position, paradoxically prefer to wait and pay 10 times or 100 times more than they would now. They don't want to jump in too early. It is much easier to carry out talks at this stage with smaller companies where the risk would be greater, because if they want to enter into something which might be a breakthrough, they need to do it sooner before the large companies snatch up the potential because they can afford it and take over control over the new therapies. So, summing up the business development activities, what we need, and Zbyszek has shown us, and this is the direction we adopted with mRNA. So we are able to move those molecules forward. We have reached levels of activity which allow us to say that they are drug-like. We have molecules which can be developed further in a typical drug discovery process. And this will improve the strong validation of the platform. Zbyszek also mentioned the screaming essays. Large companies are searching for something that will make them stand out. And this is the direction which we are developing with this in mind. And we do that by supporting ourselves using AI tools. So currently we are facing a huge challenge, but also a huge opportunity. The data that not just us, but a lot of clinical groups and clinical groups is generating and publishing clearly show that the therapeutic target we have adopted for KITE-1, that it is really important for many diseases, but most importantly in MASH and also sarcoidosis. Another element consists in the fact that our molecule works efficaciously. It changes the phenotypes of macrophagy and has potential where some drugs that have been already approved might not be effective, that is in later stages of the disease. When it comes to OATD02, we are striving to get more data and to reach our target points, that is to establish the maximum tolerated dose in patients with solid tumors. When it comes to our partnering strategy, it is simple. We are searching for a partner who is interested in an optimal continuation of our trials in sarcoidosis, which is a rare disease, but also who is ready to start a proof of concept phase 2 in MASH. As a company, as Zbyszek already mentioned, apart from the data that we have, we do not have an internal expertise for metabolic diseases. This is something we are learning very quickly, but we are still at an early stage of obtaining this expertise. So it is a steep learning curve, and this trial will be larger than for sarcoidosis, both in terms of the patient population, but also the period of administration. So it also requires greater resources. So the talks are quite advanced. For the partner to take on the responsibility and contribute its know-how in the development of metabolic diseases, in particular hepatic diseases. And in parallel to the sarcoidosis trial, the MASH trial would be started. Because each month is precious, because the survival time of a patent on the market, which will be worth many millions of dollars, each month is worth several millions of dollars. So we are fighting against time, and we hope that this arrangement on which we are working on will be successfully launched in the months to come. I think that would be all on my part, and I think that this is the last slide that we have prepared, but we will be delighted to answer any questions that you might have. Thank you so much, Marcin. Thank you for the presentation, and we are moving on to the questions from our participants.
The presentation is already available at our website under the Investors and Media tab. In the recordings, under the recordings tab, you have the PDF. That we've received several questions on OETD01, so I will ask them now. How many patients have been recruited until now in OETD01? How many are currently in the process of enrollment? I understand that this is a question for me. We are not following this enrollment at such a granular level, but the level of enrollment is still not satisfactory for us. We are still more in the beginning than in the middle of the target enrollment. Next question. Whether since OETD01 has a potential in MASH or historically in NASH, would it be also useful in obesity and diabetes treatment? Because you have mentioned the regulation of the glycolization and proteins, so maybe the Novo Nordisk or Lilly, could they be interested in OETD01? Are you in contact with them? Are you talking with those or other similar companies? I will try with the first part, preclinical part. There are specific preclinical diabetes models, and OETD01 has not been tested in those models, but it is true that the process of glycolization and also the fact that a number of persons with NASH also are suffering from obesity, but also there's those that are not suffering from obesity, but they still develop NASH. It also translates into potential. One of the publications I showed you was a correlation between the level of KART1 protein level in blood stream and its activity that correlates with insulin resistance. So there are reasons to consider that learning about different pathways activated by O001 in different disease models. We have encountered several pathways which are the same for both diseases. So they are closely related diseases to some extent, and we have to look at it translation-wise, and we have to look at the market, but we have to have strong proof to be able to start clinical studies as soon as possible. So although we see a potential, we focus on MASH for that reason. Thank you. Another question on OETD01. Is it possible to show any results in the mid 2025? If you should be able to recruit around 40 patients to be able to have the interim results by the end of 2025. Is it realistic to expect them at that time? Yes, if we take the end of 2025 as the point in time, we expect to have those results, but it will all depend on the second phase of clinical trial, where we are depending on the CROs. The predictability ends with the preclinic and the phase 1 clinical trial, where we are able to cohort patients patients, the healthy volunteers. Once we get into clinic, so we depend on true clinic hospitals, inpatient ambulatory centers, where the speed of enrollment largely depends on our researchers and clinics. So this is the plan as far as partial analysis, that it should take place by the end of 2025. All right.
And now we have a question regarding a grant for OATD01 IPF. The company mentioned that in the financial report. What doubts does NCBR indicate with regard to the OATD study? What was the total amount of co-funding, and which part regarded the IPF? I'll try to answer this. The remarks of NCBR regarded the manner of closing the project. So we are in close contract with this institution because in the recommendations they asked us to contact them, and we did that, obviously. It is quite complex because we did close the project in line with the instructions of NCBR. So right now we've been observing a pivot in the NCBR. So we are currently trying to explain the situation, to clarify it. Unfortunately, off the top of my head, I don't have the data, the figures you asked for. Disregarded the 3 projects that we're closing. This grant, from the perspective on NCBR, everything has been settled, paid for, the money was received last year. Everything from the formal financial standpoint has been closed. But what appeared was a discussion in which we are currently participating actively. Thank you. And one more question. Which amount is potentially in dispute? And when can this issue be settled? This is just the beginning of the discussion with NCBR. It's hard to tell when the discussion will end. Please excuse me, I do not remember the figures off the top of my head. I cannot answer this on the spot. But I would say it was something between 10 and 15 million Polish zloty. But we'd like you to know that they are not demanding a refund of the costs. They are asking us to resolve a situation which, from a formal standpoint, is inconvenient, because the institution simply issued two different bodies in the institution issued 2 standpoints which are contradictory. And this is the cause of the commotion, and now we are discussing with them to find a solution. So the 2 teams were recommending contact between one another, and this is what we are doing. Another question regarding OATD01. Why is the recruitment in the US going so poorly? We know that the recruitment in the EU were due to regulatory issues, but the sites in the US have been open for several months. Do you have any studies, any information, in-house information on or external information of synergy with Zetirom or a different compound which is currently in clinical trials in the MASH indication? So we have 2 completely different questions here. Yes, I read them together, but let's address the first question and then I will repeat the other one. When it comes to the first question, so when we're asking the sites in which situations patients refused to consent when offered the option of participation, they said that patients thought they were stable enough, and at this point they do not think they simply need active treatment of pulmonary sarcoidosis. In other cases, patient also said that they might be interested in participation, but the logistic itself, which is demanding because it requires a few visits in a short period of time in the sites, they thought it would coincide with their professional work. So these are the types of situations, regular ones, that we encounter regularly in phase 2 trials. To the best of my knowledge, I don't remember patients withdrawing from or refraining from joining because of the procedures in place. So what we have planned in specific consultations is quite standard. The US is not an easy place to carry out trials. It is a very prestigious location for these trials. That is why we opened our sites. However, we did not expect the majority of recruitment to take place here. Hence, our focus on European countries. As I already mentioned during the presentation, we have mapped, among others, by talking to advocacy and groups and NGOs dealing with sarcoidosis, what is the patient pathway and at which point this model patient for our clinical trials should learn about the trial. And we have identified that our previous actions are not as effective as they could be. And this is currently the element of our strategy of reformulating the recruitment support in the KITE trial, starting with the US. And the other part, the other question, do you have any in-house or external studies for combinations of your OATD01 with respitinum or a different compound which is currently undergoing clinical trials in the MASH indication? So synergistic effect. So when it comes to preclinical trial, we've tried OATD01 as monotherapy, and the result was very good. And details of the trial on MASH, as already mentioned, we are searching for a partner who has MASH in their pipeline, who has experience in conducting such trials. And currently, we are not really considering any other combination before we talk to a partner who is more experienced than us in terms of treating MASH. Let me just add that in the talks that we held, the partnering talks, and in the business development efforts, because experimentally we haven't combined those therapies, because until recently there were no approved therapies in this indication. However, most of the projects which are being developed in phase 2 or phase 3, or the one which is already on the market, that compound, they are more mechanistically metabolic, and there are few projects which would be fibrotic and which could regard the later stages of advancement of the disease. So in terms of the concept, we expect that the therapies which are being developed by numerous companies mostly are at phase 2 when it comes to the later stages of the disease. This might serve as a complementary effect. However, in terms of experiments, we haven't verified this, especially on the level of the clinical development. Most therapeutic areas currently, when it comes to developing drugs, is based first on development in monotherapy. Obviously, there are some exceptions, for example, in communicable diseases or cancer care, where from the very start, some compound might be developed as an adjuvant in combination therapy. However, MASH is a good example of a situation where drugs are being developed in monotherapy. Only when they hit the market do the scientific world use this to combine different products in different trials, and sometimes the results are quite surprising. However, this is not mainstream in drug development in commercial studies. Let me just add that similarly to the KITE trial, because I think it's a good time to underline this, currently we are looking at the inflammatory component. We are looking at the patients who are recruited. They are not patients with advanced fibrotic lesions. Once we've proven out the effect, we will be targeting patients with the later stages with the fibrotic lesions. Similarly to MASH, all our results are based on monotherapy, and the preclinical trial results indicate that there doesn't seem to be a need for a combination therapy, but that might change, obviously. Let me just add that all those questions are valid and interesting and worth considering. However, from a strategic standpoint, and the level of resources and concentration and use of resources to show something that might fundamentally change the company's situation, what we have to do is focus on something. Once we manage to sign an agreement with a partner who will take over the rights and will contribute to the company with the resources, then we can consider various scenarios, and then we can carry out various trials which might lead us towards an interesting direction, for example, combination therapy. But currently, what we have to do is to prove that the compound works in patients, and then someone needs to buy it. Because we're a small company in Poland, and we will not be able to gather the capital that would be sufficient to explore different variants and different uses of the compound. I think that in MASH, our data is strong enough to convince anyone. What we have to do is show that this also works in the clinical setting.
One more question about OETD01, and then we move to O2. What countries do you consider as backup countries? Are these non-European countries? We are considering two backup countries. Both are geographically European ones. One of them has been mentioned today, sort of between the lines, since we've had a number of talks with a number of researchers, leading some of the leading international pulmonology researchers. After the PredMed study completion, they contacted and showed interest. I mean here the Netherlands, but at this moment I wouldn't like to make any further declarations. We have at least one country, another potentially attractive country, but it doesn't mean that we are already approaching those countries. First, we have to perform a feasibility study to assess feasibility at a given country, and this is something that we're beginning now. In the Netherlands, we are starting the feasibility study, and only after we have obtained the results of the study, we will consider it seriously. But perhaps it will not be necessary to open the study in other countries. In the Kinevant competitive trial, for the similar number of 100 patients, they have launched 3 times as many sites as molecule. How many sites can you expand to within a short time? Because the the study seems to be estimated. Well, it's not a simple arithmetics because the data, the examples of Kinevant and the Millennia Resolved Lang, this is not public study results. We do not know them, but the details of it, the design of the study was presented in Vienna by the Kinevant together with the support of lead researchers. So we are aware of the lead sites in that recruitment, but it's not a simple arithmetics. It doesn't mean that one site is 3 patients, so 20 sites is 3 times 20. This is different. The number of patients may differ between sites, and the whole thing is about selecting the most prospective sites. So informally, I can tell you that the Resolve Lang study has shown that some sites are able to recruit many more patients, and some are able to recruit a small number of patients, and some can have recruited none. So it's not a question of a number, but also about the quality of the site and to lead the site properly from a responsible level. Now let us move on to the OATD02 study. In previous cohorts, there has not been there has been no toxicity. The dose has been escalated. At 20 milligrams, we have an information on no toxicity, but there is a request to add 3 more patients. Why 3 more patients and not one by one? Starting from the end, this is surely a reflection of a typical study design in oncology. It's called 3+3. There's a historical name. It's a sort of a magical number to formulate the cohort lines for those levels. Our study design provides for a certain flexibility. I don't want to comment on the recommendation of the external committee, but I think that it makes sense taking into account the data volume. It will make the data volume double in terms of pharmacodynamics and kinetics. So then we will be able to have more conclusive results, more binding conclusions, whether we should focus on the lower or higher range of variability. Yes, there is no toxicity, but we see a change in secondary line parameters of pharmacodynamics and kinetics. Another question for 02. In the context of the previous answer, I think this will also require only a short answer. What was the level of the biomarker in a patient in the 20 milligram cohort? Which biomarker do you mean? I understand that it's the arginine. I understand it's pharmacodynamic. It was variable. So the parameter was variable in different patients. So hence the decision of the committee to proceed to more patients. Arginine, again, how do you measure the arginine level? Do you measure it in circuit blood or differently? And how does it look in the OATD02 study? The arginine level seems to be a biomarker subject to level of stress, cardiological diseases, and taking taking supplements such as L-arginine. So is arginine the best biomarker to evaluate in OATD02, and is it specific enough? Arginine is a biomarker, a functional biomarker, because so far the OATD02 study has shown that if we demonstrated the therapeutic effect, it was due to the increased levels of arginine that reflected the T cells and other cytotoxic cells' ability to kill the cancer. The arginine is an exogenic amino acid. This is true. We take it in with food, but the experiments are conducted. Each patient has its own baseline, so the variability is measured against the baseline in each of the patients. So we look at the patient before and after giving and also the drug is taken before meals, so on an empty stomach. So the patient is assessed individually in that respect, and the issue of arginine in circulating blood. The most important thing is arginine in the micro environment of tumors. It would not be ethical to take biopsies regularly, but scans are done, which can show that whether the tumor has increased or decreased. And in the future, we plan on specific studies and tests that will assess the microenvironment of the tumor. But previously, the arginine level correlated with the administration of our compound, and also it correlated with the level of dose of our compound. Finally, at the end, near the end of the study, we are going to analyze the samples for other potential biomarkers to see whether there's any correlation. But today, if we look at the activity of arginase, the arginine is the best biomarker able to show whether we have the inhibition of arginase, and also arginine itself is functional as to its level in the therapeutic effect. So this is the best we have for now, for today. If I may add something, yes, of course, the arginine level is variable in different patients and in different days. So what we are interested in is our shifts in a global sort of scale. We are interested in the general movement, whether it's upwards, and not, of course, analyzing it against any standard. This is the only obvious biomarker for our drug, for which has arginase at its molecular target. So it would be difficult to design a study in which we would not try and evaluate our target, our molecular target, which is the concentration of arginine. Another comment, Zbigniew said it would not be ethical to perform biopsies. Well, they are performed in clinical oncological studies, and initially we considered such possibility, but please remember we would need a double biopsy before the start of the treatment, so baseline, and then upon the end of the study or at a selected moment of the study, going through two biopsies biopsies and invasive surgery is a high, very invasive intervention, and the majority of patients would not agree to that. So we have decided that obtaining data from very few patients will not allow us to reach our therapeutical objectives. Thank you.
Another question regarding OATD-02. How much the dispersion between and whether on the level of 20 milligrams was any therapeutic activity observed? Were there any signals of efficacy or any side effects, any adverse effects? And the question is whether this variability, the rotic acid, how did it act? Were there no increases? Let me tell you that we do not have all the results, especially with regard to the orotic acid. You can correct me, but we are currently focusing on the main readings, but the main aspect is finding the maximum tolerated dose and how the PK and PD markers they are additional information which help us take decisions. To simplify how it works, the main challenge is finding the maximum tolerated dose, and as Piotr already mentioned, we haven't observed any side effects, adverse effects, or drug-related toxicity. And when it comes to PK and PD markers, on the basis of the three patients, the variability is quite significant. For matter of precision, we can't say that there are no side effects. So there are some diverse effects which are noticed, and this is the case in all trials. So is the case in our trial. But what is of key importance are the first months of observation. So first months of toxicity observation strictly defined in the protocol, in the study protocol, and this we haven't observed at all. And this is the key driver of the study, the key premise for taking a decision on whether we should escalate to a higher dose or maybe de-escalate and go back to a lower dose. When it comes to efficacy, the possible efficacy signals on such a small sample would not allow us to make any statistical calculations. And on the other hand, given the lack of certainty as to the effective dose, which is understandable because this is standard for first in human studies in cancer care, we have to be very cautious about any signals regarding efficacy. Any such results which would suggest efficacy, any conclusions drawn on this stage would be considered speculation. So we shouldn't focus on speculation. Let's focus on the data, which we can actually corroborate. The question about the rollover in OATD-02, are there any patients at this stage of the rollover study? What was the effect of the rollover study for 2 patients, according to the data indicated in the financial report? To the best of my knowledge, currently in the rollover studies, we have no active patients. Thank you. The next question regards whether you see a realistic closure of the phase 1 OATD-02 in 2025. Yes. Yes, looking at the simple study design protocol and the time forecasts, if the escalation proceeds well, provided that the recruitment goes well, and recruitment has been going well, then yes, 25 is realistic. But this is the beauty of early phase studies. We do not know what lies ahead. We do not know whether we will be escalating the dose, whether we will stay with the currently studied dose level. And now the question regarding mRNA. Questions regarding mRNA. I will combine them. What is necessary for commercialization in the mRNA segment? What's the timeline? In the report, you state that by the end of 2025. Is it the case indeed? And the company suggested that the platform is presented to potential partners. Can we count on results of those talks this year, in 2024? So the question regards whether you intend to obtain some transaction by the end of 2024 or by the end of 2025. I think 2025. We have had many talks, and the interest has been significant. At least half of them were large pharmaceutical companies. But as already mentioned during the presentation, to move on to the later stages of conversations, the partnering talks, we need to be able to show more progress in our works. So the proof of concept itself is an important stage. It is a certain point which is a significant achievement. But companies want to see either more programs or more advanced molecules. And what we're working on, and this is our approach to identifying both the areas which are druggable in mRNA sequences, and the screening assay which will validate the molecules, which is the IP and a unique element of the platform. So to answer shortly, we're looking more at 2025. And the elements that were asked about, those follow-ups might move on to more advanced talks, which would define the terms of potential cooperation. So when we, Marcin Maurycy, went on meetings, we presented mRNA as a form of service, and we presented the tools that we have, to what stage we are able to lead a molecule, what scientific questions we can answer using our tools, our platform. And the companies wanted to know how we do it, how we stand out against the competition. Now, the two slides that we presented indicate how the molecules are evolving. So now we can change the strategy of the meetings, and now we can talk about the early stages of discovering molecules, not just talking about the platform itself. We'll see what works best. Let me combine two questions.
How does the company plan to solve the issue of further funding with the lack, taking into account the lack of grants under the last call for the FANG program, and do you plan to obtain funding for several years rather than for 1 year? So let me start with answering the question about the grants that we do not have. This is not true. We have a number of subsidies. We would like to understand better why we've been evaluated so harshly by the agency to be able to be more effective in obtaining grants in future programs, referring to the main question, which is the obtaining capital from the capital market. Well, for a biotech companies, this is undoubtedly a normal thing to apply for such funding, to look for such funding, capital funding, but this decision has not been made. But we would like to combine it with an optimal point in time that would secure the value of our programs and secure the value of our company. So this moment is surely ahead of us. Today, there's a question: why do we show those the target capital 1 year sequences? Yes, it is true that it makes things more difficult, but we want to optimize the value of our company. We want to build together with you the future of our company in such manner that we do not need. We hope that you understand us. We do not want to take a huge amount of money that we may not need, where the deal is sort of on the way or very near. From a strategic viewpoint, how can we rescale the company and drive clinical studies in the most prospective area, and how we should obtain capital? This is all connected vessels. So to make it more simple, at this moment, if we go out there to look for capital, it's to maximize and optimize the partnering transaction for OATD01, to give ourselves an opportunity to take part in a dynamically growing large market. This is the reason why we would go out and look for money that we've been able so far to obtain on a certain so-called so to say year by year basis, the sort of money that is sufficient for a shorter limited period of time, just to reach another stage of studies. If we are able to reach our main business objective for the moment, then yes, we are going to look for a larger capital, also abroad, with a prospectus, so that we have a more stable and ambitious activity, because the grant situation in Poland has never been looked so bad in our sector from the beginning of our company until now. It's not about anyone's ill will. It's just the conditions in calls of proposals, the definitions of deployment application have been created in such manner that they do not really correspond to what we're doing, on what a number of other biotech companies are doing. So I think that we see one source of funding escape us that was an important source of funding for our Polish biotech sector, starting from zero. And now, as the Biomed Association, we meet with decision makers and we try, together with other companies of the sector, to request changes that would make us eligible for, even with the most risky and innovative projects, to be able to obtain funding for such projects, and not only those predictable type of projects. It was a lengthy reply to a short question. Thank you.
We have one more question regarding the MASH trial. How many partners have you talked to regarding cooperation on a trial with OETD01 for MASH? What stage are these talks at? And how much time is needed for a decision? How much time until from a partnering decision until an application for a consent to carry out the trial? I will give the floor to Piotr, but in the last year we carried out between 20 and maybe 40 talks at different stages of development until the stage of signing the contract. We would announce it if that did happen, but we carried out various due diligence and analysis of our projects that was quite in depth. So currently we are in the process. So we have had many talks. The profile of each candidate differs. Large companies tend to wait and are ready to pay more at the end. It would be great if they paid more in the end. But to us, time is of the essence, and this is related to the need of starting the trial as soon as possible, so that with a possible success, we have enough time of the drug on the market with patent protection. Obtaining consent for the trial itself. Well, Piotr, please correct me if I'm wrong, but I think it goes much faster if we have an open R&D in the States for a given molecule and we just expand it by another indication. This is a much shorter process than starting the application process for a consent de novo for a molecule which has not received approval. I won't be objective, but I think this indication is very attractive. But if my colleagues told me, Piotr, just do it, I would jump head on into implementing such a clinical trial, obtaining the necessary consents and so on. In such a well-penetrated indication, and that's MASH. So remember that in the US we have guidelines for MASH. So the example of Resmetirom has paved the way. So other sponsors might minimize the risks so that a drug which actually is effective can demonstrate that effect in clinical trials. If we were to hold phase 2 clinical trials, the guidelines require a biopsy. So presenting on the histopathological material that the drug actually works. So you have to go to the right sites, the right countries. We have to be conscious of the significant costs and the time needed to carry out such a study. For this R drug, which recently received marketing approval, in this case, the phase 2 trial lasted 9 months. It was between 100 and 200 patients. So this is quite a significant scientific and financial venture. Thank you. And 2 last questions. They're quite short.
The company has announced 2 major publications this year. One with the Mannheim University, of which I am not sure here and now, and with Professor Daniel, and what? Well, this is a question for our reviewers. They've been keeping this publication under review for the last 2 months. I've been asking myself the same question. Yes, there is a possibility for I think with the University of Mannheim, I think this is the mass spectrometry and OATD02. I think that we would be able to publish it already, but we want to add some results from Professor Jagielski's center. So we are processing the results, and this publication is underway. And when the first one has been under review for the last 2 months, so we've been asking why does it take so long. Purely hypothetically, if you were to sell all the OATD, if you were to sell all molecules under study, would their value be larger than the Colors company capitalization at this moment? Yes, surely. The sales of even one molecule would exceed the value of capitalization. Thank you. This was the last question.